Tēnā koutou katoa. My name's Stuart Brock. I am the Deputy Vice-Chancellor Academic. It is my great pleasure to welcome you to this very special occasion to celebrate the promotion of Natalie Hughes to Professor at the University of Otago. These lectures are a highlight of my week, as I'm sure they are of many of yours. They're a wonderful opportunity for us to come together and listen to our new professors talk about their personal journeys, their research platform, and the impact of their research on others. I'd like to welcome colleagues, students and friends from across the university and beyond, including those joining us tonight by live stream. I'd like to give a special welcome to Natalie's children, Andrew and Claire, who are here in person this evening, as well as Jane, who's watching on the live stream all the way from the Netherlands, where I believe it's about 7:30 in the morning, so that's quite a commitment. I'd also like to thank Natalie's mum, June, her sisters, Tracy and Rebecca, and her uncle, Terence, who are also here in person this evening. It's not easy to become a professor at the University of Otago. In order to be promoted to that level, candidates need to show that they are outstanding in each of three categories: teaching, research, and service. Natalie has clearly met all of these criteria. It is an honour and a privilege to congratulate her this evening for that outstanding achievement, and I hope you'll join me tonight in doing the same. Natalie is Deputy Dean of the School of Pharmacy and is a pharmaceutical scientist with expertise in drug delivery, medicine formulation and pharmaceutical analyses. Her commitment to the University of Otago is long-standing, and she is an integral part of the Otago whānau. She has a Bachelor of Pharmacy and also a PhD from the university. And while she worked for a short time as a postdoctoral researcher at the University of Kansas, she returned to Otago in 1997 and has been here ever since. So thank you for that commitment. Natalie's research includes improving drug delivery for neonates, new treatments for medical health, and pain and topical treatments for promoting wound healing in dentistry. She works in multidisciplinary teams to develop new healthcare options and products for all New Zealanders. Tonight, Natalie will reflect on her academic career to tell us a little bit about her own personal journey and about the impact of her medical research on the profession, on her teaching, and, most importantly, from my perspective at least, on her students. But before Natalie does that, it's my pleasure to hand over to Carlo Mara to formally introduce Professor Hughes and give you a little more detail about her academic background. Tēnā koutou, tēnā koutou, tēnā koutou katoa.
Kia koutou. Ko Carlo Mara tako ingoa. I am Carlo Mara. I'm the Dean of the School of Pharmacy and Pharmaceutical Sciences, which Natalie is an integral part of. As you all know, friends, fellow colleagues, and students, Natalie's a very unique individual, and her role within the School of Pharmacy has been such. So Natalie, as alluded to, has been at the School of Pharmacy, when you consider, in almost every conceivable role that you can think of. So she did her undergraduate there. She did her postgraduate training there. She's been every single academic position from research fellow all the way through now to professor. So she has this really strong rooted commitment to the School of Pharmacy and has seen all the changes and all the developments that have happened over time. So she's very unique in that regard. I think she's either the second or the first longest standing member of the School of Pharmacy here at the University of Otago. The last one is sort of standing from some of the original cohorts. And it's been my absolute pleasure to work with Natalie very closely over the last 8 years. She has been my deputy dean, although she's not currently the deputy dean right now, but she's fulfilled a number of roles, including being the dean of the undergraduate programs, which we have 2 of in the School of Pharmacy right now. And Natalie has been very integral in the formation of our second degree program, the Bachelor of Pharmaceutical Sciences, which I'm sure she will talk about. Natalie was one of the first people that I met, actually, when I came to New Zealand. She took me grocery shopping when I got here, even though she was not super chuffed that I got the dean job at the time. She's told me subsequently that I was not her favorite person at that moment, but we've developed a very close friendship over time. She's forgiven me for that. So you can understand that her presentation is going to be amazing. She's had 30 years of building her story, and so I can't wait to hear this story. So I'm going to turn the mic over to Professor Hughes, or you have your own mic, and we'll take it from there, but I'm really looking forward to it.
Tēnā koutou, tēnā koutou, tēnā koutou katoa. Ko Otipoti taku kainga noho, no te awakairangi aho, ko Natalie Hughes tako ingoa. No reira, tēnā koutou, tēnā koutou, tēnā koutou katoa. Welcome, everyone. Thank you, Professor Brock, for your kind introduction and for hosting my IPL tonight. Thank you, Carlo, for your lovely introduction. You've been the Dean of the school for the last 8 years, and you've also been a friend, so I really appreciate that, and it's lovely to have you introduce me tonight. Thank you also for welcoming my close family. As I said, Jane is watching in the Netherlands, and 2 of my children are here with their close friends too, so welcome. I also see that there's some PhD graduates of mine here tonight, and I'm very privileged to see you here. You will see some of your work showcased, and you've been great to work with. Another important guest here tonight is my good friend, Professor Julia Kennedy. Thank you for travelling to be here with me tonight, and thank you for the loan of your beautiful gown. Today I want to talk in my IPL, my inaugural professorial lecture, a little bit about what I did in pharmacy. As Professor Mara has said, I've spent a lot of my time here at the University of Otago, and I chose to do that, and I do like Dunedin and Otago. I've done research in pharmacy and in formulation, and that's what I want to talk about today. But also what's important to me is the collaborations that I have been involved with and the connections with people through research. So you'll see some of that tonight. Firstly, what is drug delivery and why is it important? And also, I'll refer back to my title about molecules to medicines. So drugs are molecules. People discover them. They have activity. They do good things for people's health. But in their rawest form, they are white powders. We can't give a white powder like this to a patient. The dose is uncontrolled. We don't know how much they're going to get. It's very uncontrolled. So what has to happen is that powder, that drug substance, has to be converted into some sort of medicine, a dosage form. And you can see on this slide here, tablets, capsules, and very common dosage forms, maybe solutions of drugs for injection as well. But there are many. There are many different dosage forms available, and you probably know them, have seen them, maybe some of you have used them. And when one thinks of drug delivery a little bit more, there's no point in just having a medicine. It's got to be given to a person. So drug delivery involves making that medicine and then optimizing the dosage and the dosage regimen in the patient. Now, patients are not always humans for human medicine. Animal health. There's a lot of pharmaceutical products used in animal health as well. So patients can be people, but they can also be animals as well. So lots and lots of molecules are discovered, but only a few make it through the drug discovery pipeline to become medicines. So why is it a problem? Why do some molecules fail to become medicines? I've got up here two reasons why this might happen, and formulation scientists deal with these a lot. On this side here, I talk about permeability, and on this side, aqueous solubility. So some drugs are quite soluble, and they pass across biological membranes quite easily. Now, I'm not going to say they're easy to turn into medicines, but they're not as hard as some other potential molecules that are discovered for for use as medicines. There are poorly water-soluble drugs. These are the ones that formulation scientists need to work on and try to create strategies to help these molecules dissolve so that they can get into the body and then into the blood and then find their way to their site of action. The other category of medicines of chemicals, drugs, are the highly water-soluble ones. So these might be easy to make into solutions. They might be easy to make into medicines. But if they're poorly permeable, then they can't pass any biological membranes to get into the body. So this is drugs like insulin. They can dissolve, but we can't take that orally. We can't have tablets of insulin because they don't pass across biological membranes. When a chemical or a compound is formulated as a medicine, a dose can be given to a patient. This dose creates some sort of concentration in the body, and that has some effect on cells, tissues, and ultimately on someone's health. For example, it might be a blood pressure lowering drug. So you take the medication, it produces some concentration in the body, and that concentration has some effect on blood pressure. Now, if it doesn't have the right effect on blood pressure, what do? Change the dose. So the dose can go up and down and be adjusted to an individual. And when you change the dose, it changes the concentration, which changes the effect. The drugs have good and bad effects, so they have positive therapeutic effects, but they also have negative adverse effects. So it's a balance between those two. And dose and dosage form can sometimes have a role in getting the optimal outcomes for patients' health. So my drug delivery research kind of fits into two categories. I first look at developing new dosage forms for bioactive molecules, and then also look to getting the best out of medications that already exist. Okay. Now a little bit about me. I've looked back on my education, and I've known that I've always had an interest in science and maths from pretty early on. I went to 6 different schools. Started at Lower Hutt, went to 2 primary schools, 1 intermediate, and 1 high school. Interestingly, when I put this together, I looked at these schools and I spent the most time at Dyer Street School. So I've reconnected with that school, and I'm talking with their students tomorrow about science, maths, and technology. So it's been a positive thing looking through my background for today's lecture. My family then moved to Auckland. So I went to school in Auckland for 2 years. They then moved to Dunedin, and I went to school here for 1 year at Moreau College, which doesn't exist anymore. It's changed a couple of times, and now it's Trinity College. And it was when I was in Dunedin that I decided I wanted to go to university. I wanted to do something that had maths and science, but I wanted a profession. So pharmacy was a pretty obvious choice for me. My dad also helped me with that choice because at the end of it, I could have had a pharmacy business, and I think he quite liked the idea of that. So there were two options for studying pharmacy at the time, the University of Otago or the Central Institute of Technology in Upper Hutt. And that wasn't far from where I grew up. But I liked Dunedin, and obviously I like Dunedin because I've stayed here. And I like what it has to offer, so I chose Otago. There wasn't a health sciences first year there, but there were similar papers, similar subjects, and I actually enrolled as a pharmacy intermediate student. So I knew I was doing pharmacy from day one when I came to university. However, remembering that I liked maths, I opted to take an extra calculus paper, and I enjoyed that. Pharmacy was another 3 years of study. It was predominantly a science-based course back then, and we had a small class. So I was one of 25 in my class. I'm still good friends with most of those 25. There was another CIT, also educated pharmacists, so there were more students, more pharmacists trained there, and there was a small group trained in Otago. So after I graduated, I became an intern pharmacist. That's the first step, and I worked at Dunedin Hospital. I'd worked there in my summer holidays, and I was very happy to stay in Dunedin. One of my summer holidays, actually, I worked at Auckland Hospital, and Julia Kennedy was my supervisor, and I did a summer studentship with her, so I've had a long connection with Julia. Pharmacy at this time was located in the basement of the hospital. There were long corridors, there was a row of windows that you had to look up through to see the street. But while I was there, pharmacy moved from this location to its current site in the hospital. And this reflected the change in pharmacy and pharmacy practice becoming more visible and taking up more roles in healthcare. So I worked in respiratory and cardiology wards most, and it was then I decided I wanted to do a PhD. Dry powder inhalers were a new thing for asthma, quite commonplace now, but they were new back then. I liked compounding medicines. I liked making parenteral nutritions. So I was interested in doing a project that had pharmaceutical technology. So it was only a short walk across the road from the hospital to the pharmacy school. And I talked to the one academic at the time who could supervise such a project. And this was David Jones. So his picture is there. David. He was an academic from Queen's University Belfast in Northern Ireland. He'd recently started at Otago. It was his first academic job after graduating with his PhD. He's now the Deputy Vice-Chancellor for Education and Students at Queen's University. I started my research with him. He had a few project ideas. I picked one. This one here, right? Delivery of chlorhexidine for periodontal diseases. He introduced me to Doug Holbrook, who was a periodontist at the dental school at the time. And I learned about these things called periodontal diseases. They're gum diseases. They're caused by bacteria, and they're an inflammatory condition. And what I learned at those early meetings was that clinicians wanted better treatments. And I think that's a common story that goes through most of the work I've done. So I got started as a master's student and then upgraded to a PhD. I taught lectures and labs in pharmaceutics right from the beginning. So there was me and one other student, Sean McBride Stewart, who helped David with the teaching in pharmaceutics and drug delivery. And actually what I found, the quickest way to learn something is to teach it, because you really have to know your stuff. The other person in the slide is Professor Ian Tucker. David left during my PhD to return to Northern Ireland to a job in the pharmaceutical industry. And Ian Tucker had recently arrived at Otago from Australia to take up a newly created chair in pharmaceutical sciences. He became my PhD supervisor. He liked statistics, and I sort of started to learn a bit more about statistics, and I've kind of carried that on throughout my career. And I now have a BSc in statistics. So I did that sort of part-time while I was working for over a few years and graduated with that in 2022. David was my supervisor, and he kept an interest in the project from a distance. And he's been really an important academic mentor for me throughout my career. And it was at this time when I started my PhD, I had to think in depth about what makes up a medicine. As I said before, you've got a drug, a bioactive molecule, can't be given directly to a patient. It needs to be turned into a dosage form. Dosage forms contain the active and all other components. We call them excipients, and they control the drug release from the dosage form. Sometimes the drug needs to be released quickly to have a rapid effect, and other times it's better if it comes out more slowly and you have a slow release and a constant level of drug in the body. So drug delivery sciences help you to design materials that will achieve all of these types of release profiles. In periodontal disease, there are affected sites in the mouth, and these can be treated, so periodontists treat these sites. But the problem that was talked about at the time, or I became aware of at the time, was they could be treated, and there was a little gap or a little pocket between the tooth root and the support structures that surround it. Once it was treated, that space or pocket still stays there, it still exists. So it gets recolonized and the condition continues. So what was my solution? To create some sort of simple device. I say simple because it needs to be able to be manufactured, that could be attached to the tooth surface just at the opening of the periodontal pocket. So if the pocket was treated, bacteria were taken out, maybe we could create this little zone of plaque-free zone where there's no microorganisms and then there's no recolonization. So I say a simple device, but I did spend a lot of time creating that simple device. We made drug-eluting films. These were made of a biodegradable polymer. They had an antiseptic drug or an antibacterial drug in them. And I measured the release rate in vitro, so in little test tubes. I made a range of these, and then I was going to do a clinical trial. So I had to make some calculations and figure out what release rate I needed from my little pieces of film to actually treat the site and give a nice antibacterial concentration in the saliva film around the tooth. And I had one shot at it because I could only do one clinical trials. My calculations, I think, were good because it did seem to work. These are the tooth-bonded films. Do you see my arrow? No, you don't see my arrow. Okay. You can see the film sitting on the tooth. And when I measured the chlorhexidine concentrations, I made this little assay that could sample very tiny volumes of saliva film and measure chlorhexidine. There were anti-plaque concentrations right around the device, but a tooth away, they were low. So chlorhexidine doesn't taste very nice, so you don't really want a mouthful of chlorhexidine for days on end because it doesn't taste very nice. And having done all of my education in New Zealand, it was now time to move overseas. I got a postdoc at the University of Kansas, and we made plans to move with our 1-year-old daughter to the US. In this slide here, you can see that I did my PhD, my postdoc, and then I came back for PhD graduation. That's not normal. You usually graduate before you do postdoc. But I didn't. I like graduation ceremonies. I didn't want to miss out on it. I liked it for my family to come as well. And actually, in the last 20 years, I've been to virtually every pharmacy student graduation that we've had. So I do like graduation ceremonies. So I wasn't going to miss my own. The other thing that was important to me is my mother-in-law was the chancellor of the university at the time, and I really wanted her to confirm my degree. So I did wait, and I did get my degree when I came back. And we made it to the front page of the ODT. And I kept it, obviously, because it's here. And actually, it was a great family event. And I think Jane, my oldest child, stole the show in that photo with her beautiful hat with the flowers that were put on by my sister Tracy. So in my postdoc, was in the University of Kansas. I worked for Professor Val Stella. He was a renowned pharmaceutical scientist. It was quite daunting going to a lab like this after being in New Zealand. But I did okay. I found the skills I had from Otago were good. They were transferable and I could work well in this lab. And it was here that I learned the basics of commercialization. And the advice was, look for gaps in therapies, areas where treatments are needed, or the treatments that exist are not that good, and work on these. While doing that, keep good records of your research, because you might need them in patent disputes later on. And he was well aware of that. And this was the case because I did work on a project that had a patent dispute, and something like 10 years later, I was contacted, rang up and said, oh, can you tell us about what you did 10 years ago? And they showed me pictures of my lab book, and oh yeah, I know what I did because I learned how to write really good lab information in my books. And the date set the date for discovery of the invention that went on to be a product. So I try to tell my students that that's a really important thing to do. At Kansas, I had two roles. So I was doing pre-formulation work for novel compounds that had potential anti-cancer activity. Val's group was contracted by the National Cancer Institute. And there were two postdocs working on this when I was there. This involved things like stability, solubility work, and trying to get the compounds into a form that they could be investigated further, potentially turned into medicines. And I was lucky again here because I worked on 7 compounds, did the introductory work, so not the creative, inventive work at this stage, but one of them became a marketed drug. It's called bortezomib, and it's used to treat multiple myeloma. So I got the first lot of this in a little envelope, a little bit of drug to try and do stability and solubility work on. The other thing I worked on was cyclodextrins, which were a new excipient. They were used to enhance solubility because they were a circular compound. They were made of sugars and they made a ring. They had a hydrophobic interior and a hydrophilic exterior. So a hydrophobic drug could sit inside that molecule, and then the whole complex is soluble. So it increases solubility. We also thought it would decrease irritancy of irritant drugs, right? Because the same principle, an irritant drug is inside the cavity, and then that whole complex is non-irritant. So that was another part of my postdoc, and I enjoyed that and published in that area. So being in the middle of the US was, we were far away from mountains and the coast. It's very unlike New Zealand. So we bought an iconic American car, family car, and we went on lots of road trips through the Midwest. You can see the big skies and the straight roads. We definitely tried to get off the main roads and into some of the minor roads, and it was really a lot of fun. And of course, being in Kansas, we did go to the Wizard of Oz. This was in a little town in western Kansas, and they had all of the props from the movies there. It was really, really quite fun. Now, after my postdoc, we came back to New Zealand, and the Jeep came too. So we've had some family holidays in the Jeep around central Otago. And I worked part-time for a while. We settled back into New Zealand. And then I got my job as a lecturer. And kept going from that point. But the good thing about becoming a lecturer is I could supervise PhD students and I could have PhD projects. So that's what I wanted to do. The first of these was use the same cyclodextrin solubilizing technology that I had learned about in the US, but applied it to an animal health product. So Zime Wu did this work, and she combined a more traditional solubilization technique with the added cyclodextrin and got an injectable form of an antihelminthic drug. And when this was given to sheep, which were the target species for the product, she showed by measuring blood levels that you got an increase in both the drug itself and its metabolite. So we were able to improve its solubility, increase plasma concentrations after a single injection, and it was less irritant. So it had lots of advantages. Zima graduated in 2006, and now she's a professor at the School of Pharmacy at Auckland University. So it's nice to see her doing well. Biologic drugs were becoming quite important at this stage. They're reasonably common now, still expensive though. They're all the MAB type drugs, right? They're proteins, they're different to small molecules. So you needed different strategies to turn these into medicines. So I developed an interest in these and supervised 5 PhD students in this area. Some did fundamental work, so around stability, creating dosage forms, and some looked for more applied topics. And we're looking at delivery of growth factors to wounds, and that's what you see in the bottom part of that slide. We were able to make some devices, we were able to apply them to wounds, and we were able to see that the growth factor was retained in that wound environment and hopefully had some benefit on wound healing. I also developed a little bit of an interest in math modelling, so that sort of fitted in with my stats and maths degree that I was pursuing at the time. I had a PhD student create a mathematical model of this environment. School of Pharmacy was quite active in an organisation called the Controlled Release Society. It's an international group of academics and industrial scientists interested in drug delivery. And at one conference, we decided some of us decided at this conference to have a workshop back in New Zealand. So we did, and this was based on protein drug delivery. Proteins were new at the time, novel at the time. Students wanted to learn about them. So we could bring experts from international experts back to New Zealand, and they could interact with our students in small groups. A lot better for the students than trying to send them out to the international sites where there's lots of people to compete for the attention of these people. We brought the experts here to New Zealand, and they were able to benefit from that. And it's a really good initiative that continues today. My next big shift was to move to more clinically focused projects. So I became the head of clinical pharmacy at the school. And I was encouraged in this role by my good friend and clinical colleague, Professor Julia Kennedy. And she introduced me to David Reith, a paediatrician and clinical pharmacologist, and Matthew Zacharias, an anaesthetist. And I was invited to start working in the area of pain. Why could I do that? Because I could measure things. I'm quite a good analyst. I enjoy making assays. I can develop assays that will look at the active, so this is paracetamol in this case, but all of its metabolites as well. So when you normally take paracetamol, it gets absorbed into the body. In the liver, it's metabolized to 2 different compounds. You can see there a glucuronide and a sulfate. And that's the normal metabolic pathway for paracetamol at normal doses. As doses increase, there's a third metabolic pathway that becomes apparent. It's there with small doses, but at higher doses it becomes more apparent. This produces a very reactive intermediate compound. Can't show this one here, but this one there just on the left called NAPQI. It also can be metabolized further and detoxified to form cysteine and the mercapturate. But if it builds up, then this is the compound that binds to proteins in the liver and causes the significant toxicity of paracetamol in higher doses and overdose. So I could develop assays that could measure all of these, and we could monitor them. the trial that I was invited to work on, because I could do this analysis, was looking at the safety of 2 high-dose regimens of paracetamol in healthy adults, single-dose study. The dose, as the dose increased, the plasma concentrations were seen to increase in proportion to the dose. So the red curve is the higher dose and the blue curve is the lower dose. But when the clinical effects of these 2 doses were examined, the higher dose was no better than the lower dose. So there was no need to go that high. And there were some signs that there was some toxicity with that higher dose, or some adverse effects were starting to become apparent. This work extended a bit further into a PhD project. And this PhD was by Katie Owens. Katie's here tonight. And interestingly, there was another student, another New Zealand graduate from here, working in Cork with Julia Kennedy. And he was doing a similar type of study, and that was Philip Murphy. He was a graduate of Otago Pharmacy School. And they combined their data sets and produced a paper together on the effects of major surgery on paracetamol metabolism. And we can see the left and the right curves look a little bit different, because there is a difference before and after surgery. So it's important to know these things when drugs are used in people, in different groups of people. They may achieve different concentrations and therefore have different effects, and we might need to alter doses. I'm going to move topics a little bit here. Drug delivery presents some challenges in very young patients. So this is the work of neonates that was talked about before. A number of students have been involved in this work, which started with Catherine Sherwin's PhD. She recognised that slow infusions were very slow in little babies. They were slower than expected. And gentamicin is a drug that you need to have a high peak concentration for it to be effective. So this extended duration of delivery was probably not allowing the best to be got out of that gentamicin administration. So I, again, could measure gentamicin. So there's an assay, lots of assays with peaks. The peaks are proportional to the concentration in the sum. And we set up infusions in our lab, and we could test a lot of variables on the infusion and see how fast the drug came through the line. And surprisingly, not surprisingly, it took a long time for the drug to get through the line when it was moving very slowly. And the solution was not to give slow infusions, but to give a fast bolus into the line and let the slow line take it into the baby. So so there was a change in the way that gentamicin was given in the NICU ward at Dunedin, and that slide just at the bottom here shows peak concentrations when the drug was given really slowly on the left, and on the right when the drug was put quickly into the line. Those dots are all a lot closer together because the peak concentrations were much more reliable. So there have been several other projects in neonates, especially preterm neonates. These small patients have some unique health issues, and 2 of these are shown here. So Emma Salas on the right investigated a condition called neonatal hyperglycemia. So when a neonate's born early, so preterm, their pancreas is not well developed, so they don't produce insulin as well as an older term baby. And they experience hyperglycemia. So it's high blood glucose, and they need to be treated with insulin. So Emma's PhD looked at trying to understand this hyperglycemia in neonates, and she used a marker for the baby's natural production of insulin, C-peptide. This is a piece of the protein that's cleaved off insulin after it's as it's secreted from the pancreas. And if you measure circulating levels of C-peptide, then you know whether the insulin comes from the baby or it's from the infusion that they're given to treat their high blood glucose. The other project is Lisa Kramer. She's a neonatal pharmacist, and she knew of this problem of retinopathy of prematurity because she was a neonatal pharmacist. The treatment at the time was that the eyes had to be examined by an ophthalmologist. So the babies needed to have some medication given to put in their eyes, instilled in their eyes to open up the pupil so the ophthalmologist could see the back of the eye. But there aren't eye drops for babies. There are eye drops for adults. So large drops were being given to babies and they were flooding the eye and maybe causing some adverse effects. So Lisa's project was looking at, could we use little drops for little eyes? And she's made little drops for little eyes. Moving back to adults, again, where there's a clinical need, back to dentistry. On the left side of this slide is Sarah Hanning's PhD. She was looking at novel materials that could be used as saliva substitutes in people with xerostomia. The xerostomia is a condition where you don't have saliva produced. So it's patients have very dry mouth. People have very dry mouth if they have xerostomia, and they need to replace that saliva with something. So her project looked at what we could replace it with. Can we replace it with something better than's currently available? And she developed these frequency-dependent viscoelastic materials as saliva substitutes. They're really interesting. So I guess a mouthful there to describe those. But these materials, when you're not speaking and your mouth's at rest, they sit on the mucosal surface, so they just sit in the mouth. When you speak and talk, they begin to move. The project on the right is a bigger project. It's going back to periodontal disease where I started. And I'm working in a group with Associate Professor Dawn Coates, Warwick Duncan, 2 postdocs and 2 PhD students. And again, now we're looking at treatments for actively treating inside the pocket and using some natural products. So we know Manuka honey is good for wound healing, but honey near teeth is probably not a good idea because it's got sugar. But other products from Manuka that have antimicrobial activity could be valuable. And again, I'm looking at maybe doing some modelling of this area here to see what's an optimal release rate from our particles. So having a background in drug delivery, analytical methods, and a pharmacy background, meant I've been asked and I have contributed to a lot of different projects. And there's, so I don't miss out any of the PhD students and any of the projects that I'm involved with. I have a slide of lots of projects here. The first 2 around anticoagulant drugs. So with these thin the blood, stop the blood clotting. But dose is important because if you give too much, the adverse effect might be bleeding. If you don't give enough, then the patient has risk, or the person has risk of clots forming. So there's a really narrow balance in dosing for these agents. Lucy's looking at information given to people about their medicines, and here about drugs used in mental health for pregnant women, so they can make good decisions about or informed decisions about whether to continue or change medications. Paracetamol story continues with Gianna Salas's PhD, and she's looking at pharmacokinetics and how that changes if children have fever. This one here, fact and fiction communicating science through crime fiction. I'm not 100% sure how I got involved in that project, but I did. Yeah. the initial project, this was for Van der Simon's PhD, there was a poison in the crime fiction novel. And I did want to learn something about science communication, so I agreed to be a supervisor in this project. And I said about patients not always being humans, sometimes being animals. Arlene McDowell has a really good interest and experience in wildlife, dosing wildlife in medicines for wildlife. I'm hoping to help with some analysis of some of those samples so we can see how species like penguins metabolize drugs. I talked about commercialization right at the beginning. I learned about this when I was in Kansas, and when I came back, I was always looking for an opportunity to be able to do this, and I luckily found one with this project. So all drug delivery scientists want to work on projects that eventually become medicines. Ketamine is an old drug that's finding new uses in mental health. And I learned about this when I met Professor Paul Glue, and again, was looking for someone to measure some samples. So I get invited to a number of projects through that avenue. He'd done a lot of work on ketamine and treatment-resistant depression. When I joined, we were looking at whether ketamine also had beneficial effects in anxiety. And in one of these studies, we did take blood samples from participants, develop an assay, and measure the pharmacokinetics of ketamine and its active metabolite, norketamine. All these studies were done with injectable ketamine. Is that the best drug delivery solution for ketamine? Probably not. Injections not very convenient for patients. So Paul asked me if I could make tablets. And I said, yes, I know how to make tablets, but I can't do them in the quality that you need. I can't make them in the quality or the scale that you need. We need an industrial partner. And I had some industrial contacts through pharmacy and drug delivery. And we met with Peter Sermon from Douglas Pharmaceuticals when he was down for a conference in Dunedin. We had a nice meeting. He liked the idea. We all went up. We then joined with Alex Tickle, and we all went up to Auckland to pitch this idea at Douglas. And they seemed to like the idea and pursued the project, and we've had a really good collaboration with Douglas Pharmaceuticals. And they have made an oral ketamine dosage form. These are tablets. There's a controlled release. They're released slowly over 12 hours. When given to healthy volunteers, we measured plasma concentrations of ketamine and the active ketamine and its active metabolite norketamine. And because ketamine is when given orally, it gets absorbed and passes through the liver metabolizes it. And actually what we see in these graphs is that the higher curves are actually the metabolite norketamine. So we have lower levels of ketamine, and maybe that helps the tolerability of this medication in patients. Phase 2 studies have been completed by Douglas Pharmaceuticals. And if we think about what the drug delivery pipeline looks like, it's very long, right? Pre-discovery. There's some preclinical work. The clinical trials are phase 1, 2, 3, and then there's a review and regulatory approval. So it is a long process. So with this product, controlled release ketamine is finished at phase 2. So it's getting there, and it's looking promising from those press releases, which is really exciting. So we continue to do some work in this area. I'm making better assays, which is what I do. The previous assay was able to measure norketamine and ketamine. We now have a ability to measure a potentially active third metabolite. That took us a wee while, and I think Feroz is in the audience. He's worked very hard on developing this assay. Work also continues to find out if ketamine is helpful in other mental health conditions. Also on biomarkers for ketamine's effect to know if it's working or not. And alongside these, we will be measuring plasma concentrations of ketamine, norketamine, and hydroxynorketamine now, and see if these correlate with the biomarkers and the clinical improvement scores. I like to try and invent things. I like the proof of concept rounds that Otago Innovation run. I've been to a number of them. Pitching ideas is quite fun. You have to say, give your idea quite a quick pitch to see if someone is interested in taking this further. We did win this at 1 year. We had an idea for pain relief research, and we're working on it. It's yet to come. So I've done a lot of things in drug delivery. I thought I'd just sum up by saying, what do I know now? I know neonatal infusion lines are different to those in adults. I know little eyes need little drops. I know pharmacokinetic analyses linked with clinical measurement of clinical effects can help guide dosing in different patient groups. I know gum diseases need local treatments. We all need saliva, and we need better improved saliva substitutes. The lower peak concentrations and sustained levels of ketamine seem to help its tolerability as a therapeutic agent. I didn't talk too much about enantiomers, but enantiomers bind differently to receptors, and that possibly modifies their effects and can have advantages. Protein bioactives need stabilization in drug delivery systems. Drug solubility and stability are key ingredients to medicines being developable. Learning about PK in wildlife could help support their dosing decisions. Commercialization's a really long game, and crime fiction's better if science is portrayed correctly. So I really, I've talked or skimmed through, gone through quickly PhD projects, and I'd like to acknowledge the PhD students who do that work. I've enjoyed working with you. I love seeing what you do after your PhD. It's a very rewarding part of the job. So thank you. I've worked with many other students as well. So lots of students through the lab on various projects. And a group of students from the University of Bath used to come here for elective projects. It's quite a big group of students now. It's nice to see what they're doing as well. We also have a number of students from Copenhagen doing masters here at Otago, and that's been a very good collaboration. I want to acknowledge collaborators within New Zealand in the pharmacy school. I've worked with a lot of people. I've been here a long time. I've worked with just about everybody. And I've worked with a lot of people at the University of Otago, but also internationally. So New Zealand is quite a long way away from the rest of the world, so we do have to travel. And I'm grateful for the patience of my children who put up with my travel. You do have to travel, and you do have to make connections to be able to do research. I want to acknowledge research funding. Of course, we can't do research without funding. And especially the Pharmaceutical Society of New Zealand, who started me off with a PhD scholarship and allowed me to have this career. Need to acknowledge study participants and their whānau, because without their active and generous participation, we wouldn't have anything to talk about here. Staff and students of the School of Pharmacy, there are many over the years I've been there, and I probably have been there the longest now, Carlos. And I kind of want to end with this, because as Associate Dean for undergraduate programs, I've done this role on and off for probably 15 years. And I've been at the School of Pharmacy for 23 years as an academic. There's an amazing amount of work that goes on in curriculum development. The program that I had many years ago is very different to what we have now. We still have a really strong science base, and we work hard to make sure that stays. But we prepare our graduates to be key members of the healthcare team. So they have a lot more clinical, a lot more patient-facing education. We've introduced, as was said at the beginning, a degree called the Bachelor of Pharmaceutical Sciences. We're the only university in New Zealand to offer this degree. And we educate students through this degree for the pharmaceutical, biotech and related industries. So my slide goes science for pharmacists and scientists for industry. I think having these two degrees co-located in the School of Pharmacy allows us to gain strength as a school. So we have both. Lastly, I acknowledge my family, my close family. The kids have grown up alongside my academic career. Acknowledge my dad, who encouraged me to go to pharmacy. He probably didn't think I wanted to do this, but that was great. And my sisters, and I'm very pleased that they were able to make it here today to hear a little bit about what I've done. And I still like road trips. We've got a road trip there. I made the kids come with me. So, ko te pai tawhiti, whaia ki tata, ko te pai tata, whakamoa kia tina. Thank you, everyone.
Thank you so much, Professor Hughes. That was really enjoyable to see how far you've come. And that one slide, which gives the whole range of activities that you've done over your career, is amazing. Just the breadth of things from crime fiction all the way from, doing pharmacokinetics in penguins. So that is quite the span of research. I think that is absolutely fantastic. There's a small gift from the university. I don't know if anybody wants a photo on. Photo on?
Yes, photos. We used to doing this photo on.
Oh. I'm just going to turn it over now to Professor Megan Gibbons for some brief words.
Kia ora, thank you. So I'm the Pro Vice Chancellor of Health Sciences, and it's my absolute pleasure to listen to Natalie tonight and hear her journey and where she's come and where she is now. So congratulations. And I'm going to thank you all for joining us tonight and welcome you to come and join us at the Staff Club for refreshments now and celebrate Natalie's success. So thank you very much.